Think that only one moment you're alive, breathing, thinking, feeling, and the next, everything stops. Your heart, your breath, your brain, all go silent. But what happens after that silence begins? Does the body just die instantly? Or is there a hidden process still happening inside? In this videocast, we'll take a simple, science-based journey through what really happens to your body after death, step by step. If you're interested in this topic and are really curious to know, come with me.
Today we're exploring a subject that, well, it's usually discussed in whispers, isn't it? Shrouded in ritual, and you rarely get the sharp, objective lens of science on it.
Right.
What actually happens to the human body right after clinical death and, in the hours and days that follow.
It's a really profound question, and our sources, they give us a truly unexpected answer, actually.
Yeah.
Our mission today is sort of to transform this topic, which, let's face it, many find frightening,
Sure.
into a fascinating roadmap, a journey of biological and molecular change. And I think the first, maybe the most crucial takeaway is that death isn't just one single instantaneous thing.
Right, not like flipping a switch.
Exactly. It's a slow, methodical, staged process.
I think that shift in perception is key, isn't it? Going from thinking of it as instant to seeing it as this delayed chemical cascade. That's really what we're diving into.
Yeah.
And to help visualize this, there was that analogy in our research, the body as a sophisticated factory.
I like that one.
And clinical death is like the main power suddenly being cut.
Exactly. The big machines, the heart, the lungs, the conscious brain, they stop immediately. But, the whole building doesn't just go dark all at once.
Ah, okay.
There's residual heat, residual power, little chemical processes keep going for a bit.
Okay.
Then eventually the internal maintenance crew, in this case, the bacteria, they come in to start the complete dismantling.
Okay, before we even start that scientific clock, here's the fact that just absolutely stopped me cold when I was reading the sources.
Yeah, which one?
We have scientific evidence that some genes in the body, keep working and can even turn back on hours after clinical death.
It's true. It really is mind-bending. It's what science calls the thanatotranscriptome.
Thanatotranscriptome. Wow.
Yeah. And if you think about it, this one fact just throws a huge wrench into our usual, binary definition of life and death.
Totally.
If the molecular machinery is still getting commands, still executing them, well, where do you draw that final line?
That is the mind-bending question. We'll definitely circle back to that. But first, let's establish the starting line. We need to be clear on what clinical death actually is.
Right. So traditionally, clinical death is defined when those vital functions stop, the ones that sustain the whole organism.
Like the heart stopping.
Yeah, the heart stops pumping blood. So that critical supply line of oxygen to all the tissues, it just shuts down.
And the lungs stop breathing.
Right. No more gas exchange.
So, okay, no oxygen means the brain, the cells. They quickly stop their main jobs, and the vital signs just disappear. That's the classic lights out moment we think of.
Correct. That's the traditional view. But here's the critical nuance, the thing we need to grasp.
Okay.
While the whole machine has stopped moving, the individual parts, the trillions of cells, they aren't all perfectly synchronized.
Ah, I see.
The body's ability to keep everything stable, homeostasis, that fails immediately. But the cellular processes, they run on leftover energy, internal enzymes. They keep going until those resources are totally gone.
And those processes that keep going, they're really dependent on what's happening outside the body, right? The environment.
Well, absolutely. Think about a car engine cooling down after you turn it off.
Yeah.
How fast it cools depends entirely on the surroundings. Is it hot out? Cold? Is there a breeze? Same for the body. Temperature, humidity, clothing, even if the body's in water. All these things act like accelerators or brakes on everything that happens next.
Okay, so the lights are officially out, heart stopped. What's the first visible sign the body gives us that this chemical cascade has actually begun? Let's get into those early changes, the mortises.
Right, the mortises. The very first one, noticeable literally within minutes of the circulation stopping, is pallor mortis.
Paleness.
Exactly, paleness of death. Since the blood isn't being actively pushed through the tiny capillaries anymore, the skin, especially in the areas facing upwards, loses its color really fast.
And that quickly leads into one of the really identifiable markers, doesn't it? Livor mortis. Lividity.
Yes, livor mortis starts pretty much as soon as the blood stops moving. Gravity just takes over.
Right.
It pulls the blood down into the lowest parts of the body. Since there's no pressure keeping it flowing, it just pools there.
Okay.
And that leads to this discoloration, deep purple, maybe bluish patches on the parts of the body closest to the ground. It starts within an hour or so.
And it becomes fixed, right?
Yeah.
That's important for forensics.
Crucial. It becomes completely fixed, meaning it won't turn white if you press on it within about 12 hours. That fixation tells investigators if a body might have been moved after death.
That makes total sense. It's like a physical map of gravity's effect at that time.
Mm.
Okay, next up. The body starts losing heat. Algor mortis.
Algor mortis, yeah. That's simply the body cooling down from its core temperature around 37 Celsius until it matches the temperature of the air around it.
And the rate isn't simple, is it?
Oh, it's highly studied but notoriously tricky. Generally, they say about 1.5 to 2 degrees Fahrenheit per hour at first, then it slows down. But again, a heavy blanket lying on cold concrete that can drastically change the cooling curve.
Okay. Now let's talk about maybe the most dramatic early change.
Yeah.
Rigor mortis, the stiffness. This one always seems a bit counterintuitive.
Yeah.
Why do muscles suddenly lock up like that?
This is one of the most fascinating chemical bits, I think. To get rigor, you first have to understand how muscles work when you're alive.
Right.
Your muscle fibers contract and relax using ATP adenosine triphosphate. That's the cell's energy money, basically.
Hey, the fuel.
And relaxation specifically needs a fresh supply of ATP to actively unhook the tiny filaments inside the muscle fibers.
And ATP needs oxygen to be made, which is now gone.
Precisely. Once the oxygen supply stops, ATP production just ceases.
Yeah.
But the process of muscle contraction, which involves calcium flooding into the fibers, that might have already started or be ongoing. So without ATP to pump that calcium back out and actively force the muscle fibers to release, they stay locked together. It's like, well, it's essentially a permanent, chemically enforced cramp.
Like clenching your fists super tight and then just being stuck like that.
Exactly that, stuck in contraction.
And usually that stiffness starts, 2 to 6 hours after death, peaks around 12 hours, and then it fades away. Why does it fade?
It fades because of the next major process kicking in, autolysis.
Yeah.
The actual cell structures themselves begin to break down. Rigor is temporary. It's just a chemical reaction depending on the muscle structure being intact. Once the muscle tissue itself starts to really decompose, maybe around 36 to 48 hours later, the stiffness just goes away.
Let's talk more about autolysis, self-digestion. That sounds, like something that should only happen inside your digestive system.
That's exactly what it is, but happening inside every cell. See, every cell has enzymes locked up inside little bags called lysosomes. While the cell's alive, those membranes are fine, the enzymes are safely contained. But when the cell dies from lack of oxygen, those membranes fail.
Uh-oh.
Yeah, those powerful digestive enzymes spill out and they immediately start breaking down the proteins and carbohydrates of their own cell. It's like internal chemical demolition.
Wow.
And interestingly, organs that are really rich in enzymes, think liver, pancreas, they're usually the first places you see signs of this self-digestion.
Okay, so autolysis sets the stage perfectly for section 3, right? The deep breakdown phase where the microbes take over. Decomposition.
Right. Once the body's own defenses are down and autolysis is well underway, the whole ecosystem just shifts completely. Putrefaction takes over.
Putrefaction, okay.
That is mainly driven by the trillions of bacteria, especially the ones that normally live in our gut, like Clostridium. When we're alive, they're helpful, symbiotic even.
Skipping digestion.
Exactly. But now, they're unleashed.
The buffet is open, as you said earlier.
Pretty much. They start digesting tissues, releasing huge amounts of metabolic byproducts, mostly gases, methane, hydrogen sulfide.
Ah, that explains the bloating.
Yes, that causes the significant bloating and distortion of the body. And it also produces those complex, very strong odors associated with decay.
You mentioned odors. Our research pointed out that the body releases hundreds of chemical compounds during this phase.
It does. Over 400 volatile organic compounds, VOCs. These are basically the chemical signatures of death.
And forensics uses this.
Increasingly, yes. Scientists are using electronic noses to try and identify these specific chemical cocktails because the mix of VOCs changes pretty predictably over time.
So it gives them a better time of death estimate.
Potentially, yes. A much more accurate window than just relying on body temperature or rigor alone.
That's a really powerful application.
Yeah.
But let's circle back now to what you called the most revolutionary finding, that molecular echo, the thanatotranscriptome.
Ah, yes. This is where things get truly weird and fascinating. We used to assume pretty much all molecular activity just stopped when the heart stopped.
Right.
But scientists studying gene activity after death found something amazing. Not only did gene degradation happen slowly, but certain genes, sometimes nicknamed zombie genes.
Zombie
Yeah.
genes. Seriously.
Well, yeah, colloquially. They actually ramped up production of mRNA for up to 48 hours after clinical death in some studies.
But why? Why would genes suddenly switch on after the organism is dead? What are they trying to achieve?
That's the big question. Many of the genes that activate seem to be related to stress response, immunity, and surprisingly, even fetal development pathways.
Fetal development.
Yeah. The current thinking is that as cells get starved of oxygen and everything goes wrong, they launch this massive, final, almost frantic effort to repair themselves or maybe even restart, like reverting to some kind of emergency programming.
Sending out distress signals even though the main system is already gone.
Kind of like that, yes, even though the larger organism is beyond saving.
That is just profound. If genetic instructions are still being read and acted on, it creates this biological window, maybe up to 2 days, where we really have to rethink what dead means at the cellular or tissue level.
And think about the practical side of that, organ transplants.
Right, viability.
Exactly. If we can figure out precisely which genes stay active and for how long, we might be able to extend the time window we have to harvest and transplant viable organs and tissues. Things like skin, bone, corneas, perhaps.
That would be huge. Okay, so after all that internal chemical warfare and the bacterial breakdown runs its course, we eventually reach the final physical stage, skeletonization.
That's really just the point where all the soft tissues have decomposed or been removed somehow, leaving only the hard, bony structure.
And the time for that varies wildly.
Hugely variable, more than any other stage. It could be weeks, it could be many, many years. It depends so much on whether the body was exposed to air, buried deep in a dry place, a wet place, scavengers involved, countless factors.
Okay, let's shift gears a bit for this last section to perhaps the most personal aspect, those final seconds of consciousness. Our sources touched on research looking at brain activity right around the time the heart stops.
Yes, this is really cutting-edge neuroscience. There have been studies monitoring patients, unfortunately, during cardiac arrest, and they've found some astonishing patterns.
Likewise.
In some cases, they recorded brainwave patterns that look very similar to what you see during dreaming or maybe intense memory recall. Really high-level activity, specifically gamma waves.
Gamma waves.
Yeah, intense brain activity in the seconds leading up to and even immediately following the heart stopping.
And that specific finding from our research brainwaves detected up to 30 seconds after the heart stopped. In one case, that seems major.
It's a critical data point, absolutely. 30 seconds of detectable patterned neural activity after the circulation has failed. That really challenges the idea of an instant cognitive blackout.
Right.
And this lingering activity is being scientifically linked now to explaining those common features of near-death experiences, or NDEs.
You mean people reporting floating, seeing bright lights, life flashing before their eyes?
Exactly. Those reports aren't necessarily proof of something metaphysical. They could be the brain's final, intense burst of activity as it tries to make sense of the sudden catastrophic loss of oxygen.
So consciousness might actually linger for, 20 or 30 seconds?
That's the scientific theory being explored, possibly sustained by just residual chemical energy before all electrical activity finally fades. It's ongoing research, but it's definitely reshaping how we think about the exact moment the brain goes irreversibly silent.
Beyond just those final moments, though, this whole understanding of the post-mortem journey has really powerful uses, doesn't it? Especially in forensics.
Oh, it's fundamental to forensic investigation. We mentioned those 400 VOCs.
Right, the chemical signatures.
Being able to analyze that signature helps investigators get much better time of death estimates than just, say, checking body temp. And knowing the timing of rigor mortis setting in and fading, or livor mortis becoming fixed, that provides hard data points about when death occurred and if the body was moved.
What about those strange little movements sometimes reported after death? Are those related to these chemical processes?
They are, but they're absolutely not signs of life returning or anything like that. They're simple biophysical responses.
Okay.
Sometimes limbs or fingers might twitch slightly as muscle fibers relax unevenly or as tissues dry out and contract or just shifts in tension. It's purely passive mechanics, no nerve signals involved.
Gotcha. And finally, that fact about eye patterns was surprising too. Iris recognition.
Yes. Iris recognition technology relies on the incredibly unique and complex patterns in the iris of your eye. Unlike fingerprints, these patterns are protected inside the eye, and they remain surprisingly stable and identifiable for hours, maybe even days after death.
So another durable forensic tool for identification. Okay, let's try and wrap this deep dive up. I think the core realization, the thing that came through in every source we looked at, is that death isn't just an endpoint. It's a really complicated journey, a staged, predictable, and frankly, surprisingly active process that stretches way beyond the simple moment the heart stops beating.
And all this knowledge, it leaves us with some really profound scientific ambiguities, doesn't it?
Oh, so?
Well, discovering the thanatotranscriptome, seeing those lingering brainwaves, it forces us to ask, when, biologically speaking, does irreversible death actually happen?
Is it when the heart stops?
Yeah.
When the brain goes silent? Or maybe when that last zombie gene finally switches off 48 hours later?
Right. And the answer has direct impacts on medical protocols, on organ transplant viability science. Yeah. It matters.
Science can measure all these things, can it?
Right.
The energy drain, the genetic echoes, those final electrical flickers in the brain. But the biggest question this whole journey raises, I think, is the one that remains just beyond the lab's reach. If science can map out the beginning, middle, and end of the body's physical and chemical existence, is there any way to scientifically prove or disprove the existence of something like a spiritual consciousness, a soul maybe, that's separate from all that biology?
Well, the scientific data defines this incredible journey of the physical body into stillness. It's magnificent in its own way. But for now, you're right. The answers to what, if anything, happens beyond that chemical endpoint, they remain, maybe beautifully, maybe powerfully, in the realm of philosophy and faith in our human stories.
Now you know. Death isn't just one single moment. It's a slow, fascinating process where your body transforms, cell by cell, back into nature itself. From the final heartbeat to the quiet work of bacteria and time, it's all part of life's natural cycle. So next time you think about death, maybe you'll see it a little differently. Not as an end, but as a transformation. I'm really glad you joined me in this videocast. If you found it interesting, don't forget to like and subscribe. There's so much more science to explore together.